protein structure. Let's go. Okay, so this is adapted from a colleague at the University of Reading in the UK, and she's very good at this, which is why we're going to use some of her slides. Shown at the top is basically the problem that proteins run into. So it's possible to rotate around pretty much any of the bonds that are there, and there are different names for the bonds. We will talk about those. It'll be fine. However, certain rotations, depending on which amino acid we're talking about, are going to produce clashes. And every time you see that big sort of red and yellow thing going on up there, that is a steric clash. That's two atoms being too close to each other. And so you can think of them as being unhappy, or you can think of them as, repelling each other, basically, which is more correct, I would say. And so that, in principle, limits the possible ways that a protein can actually fold. Because every protein is going to be made of peptide bonds. And so what you're looking at up here are peptide bonds with the red dot, I believe, being the what they call the side chain, the part that makes each of these different. And depending on which one you're talking about, they will have more or less freedom of movement for each amino acid. And by understanding that, we can actually take a string of amino acids, and there are only certain ways that this will be allowed by the laws of physics to actually fold up and get nice and close to each other. And usually somewhere within those ways is where you will find the actual, honest-to-goodness protein structure. It'll be, more or less the path of least resistance, or at least something very close to that. All right, let's talk about it. So we're going to talk about, ways to visualize protein structures a little bit. We'll do more of that later. Understand the peptide bond big time and study the Ramachandran plot. That's going to be in this video. Then we're going to have another video after that's going to talk about protein primary, secondary, tertiary, quaternary structure, and we'll tell you what that means. And the common elements that you find. Because like with the little diagram above, even though a protein in theory could adapt all sorts of weird twisty conformations where you'd never be able to predict it, it's very limited. And there are only certain combinations that will become stable, that will be able to hydrogen bond, for example, with other parts of the molecule and basically lock it into position. And so proteins are like little machines, and they're able to do their job because they adopt a particular shape, what we call a protein conformation. And that protein conformation kind of tells you what the protein does, what it is, where it came from. Yeah, it's really neat. Okay, so let's get into it. All right, jump over here. There we go. Okay, proteins made of amino acids. You're going to learn these amino acids, or rather you're going to learn their types and their single letter amino acid codes, because we're going to be doing so much with single amino acids in this course that it's just, it's going to take 5 minutes off your learning curve every time we have to talk about one of these. So we're going to get it out of the way at the beginning. So that's the first thing we're going to do. So these are the amino acids. These are the rough categories that you can put them into. So valine, and we're categorizing them so all amino acids have the same backbone, but they have different, what we call side chains. And so the backbone is shown down here at the bottom. It's got a nitrogen at one end and it's got a carboxyl at the other end. So it's got an amine end and a carboxyl end. And in the middle, it has a carbon that's going to be attached to something. Could be the smallest one with just 2 hydrogens there, basically no side chain to speak of. Could be one of these giant ones. Like look at arginine. It's got this big long chain. So it's got 3 extra carbons there. And then 3 nitrogens around the carbon. It carries a positive charge. The environments where you will find arginine are going to be very different from the environments where you find glycine. And the properties of arginine are going to be very different. It's kind of like, I think of it as like a charm bracelet. Do you know what I mean? It's like a little bracelet you can wear that has different little shapes you can put on it, and whatever, people customize them around their life. only this is what the protein looks like. It's a little string, and it has a bunch of little side chains. So those are like our charms that hang down. If you get the right side chains next to each other, they can actually do chemistry. This is where enzymes come from. So oftentimes it's going to be some of these positively charged ones, or there are a couple here that can have a negative charge sometimes. The acidic amino acids up here. There are others that will have potentially reactive oxygen on them. So something like a serine or a threonine. And some others that will have a potentially reactive cysteine. It can react with another cysteine, or it can actually do some chemistry. So you'll find these particular amino acids in key positions. They'll occur elsewhere, but the key position is always going to be a couple of these things, because they're able to attack bonds, chemical bonds, and break them and make them and move things around. And they can support each other. So you'll see enzymes where a serine is supported by a histidine, and the histidine, which is down here, is then going to be supported by something like an aspartic acid. And so aspartic acid is going to be up here. And they're going to be linked together in such a way that they can trade electrical charges, they can trade hydrogen ions, and that's what's going to make an enzyme work. So your job is going to be to learn the amino acids. I want you to know the single letter code. So like glycine is G. For most of them, it makes sense, but there are a couple that start with L. So like leucine is L, but lysine with a Y, where is that? Is going to be K. There it is on the bottom row. Yeah. There's some that you can remember with stupid mnemonics. Tryptophan, if you say it like twiptophan, is what I was told, you'll remember it. And I remember it. I still remember it. Phenylalanine makes a F sound, and so you start it with an F. F is the single letter code. So once you have that memorized, I want you to know what amino acid it is, what its single letter code is, and which category it falls into. Now, there are a couple of these that potentially fall into multiple categories, and we can get into that. Something like a tyrosine has both a nice super duper hydrophobic aromatic ring, which is not going to want to be anywhere near water, but it also has a hydroxyl on the end, which is occasionally going to be reactive. A lot of phosphate groups are going to be added to these free hydroxyls on the end. So that's where we're going to start. That's your little assignment. A little bit of protein, amino acid anyway, memorization. Now, what are we not going to learn? Well, you're not going to have to learn the entire genetic code wheel. It's not that hard. It's just like another step beyond knowing your amino acids. Because, for example, G followed by G followed by anything is going to be the only way to spell glycine. So it's GG, and then any nucleotide you could say is N. GGN, it's a glycine, and so on. You can do that for most of these, and there are only 20 amino acids, and so there's really not that much to learn. But we're not going to do that. You may need that if you go into synthetic biology. It might make your life easier, but I don't know where you're going to go, and so we're doing what you need to actually get through the course. That's going to be what we require, because that seems to make sense, at least to me. All right, so that's where we're going to end this little part, and then we'll actually go into the basics of the two peptide, the bonds that you find in a peptide, in a protein. All right, see you next video.